Hello and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac. Hello, everyone.
How are you doing, Andy?
Good, thanks.
We're talking this week about antibody-drug conjugates, or ADCs as they're known in the trade. Like an awful lot of drug modalities, they've been around for a while. They've had a very checkered history. There's been the usual ups and downs, but you could argue this year it's been a bit of a sort of a feeding frenzy. Some big acquisition deals, which we can talk about, and also quite a number of hefty licensing deals. And the list of approved ADC drugs is steadily growing after a relatively slow start. There's been no ADC approval this year, actually, funnily enough, and there was only one last year, but there was quite a few in 21 and 20 and 19. But obviously, the original of the species has a certain notoriety attached to it, Mylotarg. Gemtuzumab ozogamicin, I believe, is
Catchy.
it's known to its familiars. Developed by Pfizer and Celtech, which is kind of going back a bit, were involved way back when. First approved in the year 2000, taken off the market about 10 years later or so, because of a combination of the fact that it was toxic, lacked efficacy, and unfortunately was killing people without necessarily giving them any kind of a therapeutic benefit, which is it was a pretty long time on the market for such a bad drug. And it popped back up again in 2017. A new dose regimen that was developed showed that you could get efficacy and much better safety, and Pfizer is back in business. It's a very strange story, isn't it, really?
It is, and it sort of highlights the components and also the progression of antibody-drug conjugate technology. So, the three components are obviously an antibody that finds the right antigen on, in most cases, the cancer cell. There's the toxic payload, which is traditionally or typically a cancer chemotherapeutic agent or chemotherapy that's too toxic to be used systemically.
Yeah.
And then there's the linker, and that was the reason, the weakness of the linker that resulted in the rise and then fall of Mylotarg, and largely the clinical and commercial profile of all other antibody-drug conjugates, because it's a tricky line to play. You need something that sticks the payload to the antibody. So when the antibody finds the antigen on a tumor cell, then it releases it there, or it's phagocytosed and it's taken into the cell and it releases it there.
Yeah.
But those release characteristics, until the last 4 or 5 years, have not been able to be as modulated as much as they have done in most recent ones. So Mylotarg, the toxin, was released too early. It's possible there are some that tried experimentally that never released them at all at the right place. So it's that linker chemistry that's advanced the field.
And there was also the DAR, the drug antibody ratio, is another very important parameter, which I didn't realize until an honest person revealed the dirty secrets of ADCs to me at a conference, that it can be highly varied in the early development efforts. So that one antibody might carry 2 or 4 copies of the cytotoxic payload, another might carry 8, and therefore you get a drug product that is not a consistent drug product. There's got a lot of variation within it.
Yeah.
That can therefore, if you can't control that, you have a real problem on your hands, I think, if you're a pharmaceutical product manufacturer.
Yeah, and again, it also emphasizes the progression of linker technology, because we've got to that stage where, and you think even second generation antibody-drug conjugates like Roche's Kadcyla, which is an antibody-drug conjugate of its first generation naked antibody Herceptin for breast cancer. I remember someone, a business development in a pharma company, told me once that the loading of the DAR, drug antibody ratio, and even the position of the payload on Roche's Kadcyla, it's largely random, which is why, particularly with that antibody-drug conjugate, they quote a mean drug antibody ratio, but the standard deviation
Yeah.
is quite high. You look at Enhertu, and it goes up from the average of 2.5, I think, for
Yeah.
with Kadcyla to 8, but the standard deviation is much lower than with Kadcyla. And you've got to think, if you're sticking on, or if your chemistry linker technology is enabling you to put a payload onto an antibody, sometimes with Kadcyla it can go virtually anywhere, and it could go in the wrong place. It could go in the regions that need to bind the antigen, so that actually doesn't do its job. But higher and more reproducible drug antibody ratios are another facet of what improved linker chemistry has brought us.
And advances in antibody engineering means that people are now able to do site-specific conjugation, or at least they claim that they can do this. So subsequent generations, most of which are probably still in the clinical trials, will be better defined products with therefore more predictable safety and efficacy characteristics. To me, it's been sort of an aspect of this drug class that, I mean, there's no great cover up here, obviously. Everybody who understands these things understands that's the reality. But when they're being talked about in kind of non-technical contexts, you think that these are settled products, and they're not settled products, because you can have what you think is a product, but it actually has an array of characteristics because it contains an array of different molecules within the actual drug dose. And I think that's been one of the reasons why they haven't been a total slam dunk. Because again, as is the case with so many concepts in biotechnology, this idea has been around almost since the first monoclonal antibodies were invented in the mid-70s. People have been talking about this for a long time. It took a very long time to even get this half right. You could say it was Mylotarg. But they're getting it right now. And as is the case in any sphere of innovation, it's not one single breakthrough, it's progress on multiple fronts. Antibody engineering, conjugation, linker chemistry, better understanding of epitope binding as well. Very, very important. And yet, when we look at the efficacy of the products that are out there on the market, I haven't looked in any huge detail. I did take a quick look at Enhertu, which I suppose is the big product, that kind of the big, huge deal between AstraZeneca and Daiichi Sankyo from a couple of years back, involving something like over $8 billion eventually in milestone payments, including a huge whopping, what was it, 1.35 or something billion up front. Big numbers. Huge expectations attached to this drug. And so far this year, it's done $1.8 billion in sales. That's over the first 3 quarters. So it'll presumably comfortably break through the $2 billion threshold this year. And, you know, there's sales forecasts of $10 billion plus attached to it across multiple solid tumor indications. But it ain't a curative drug. I mean, it's certainly better in clinical trials than either its predecessor, you could argue its competitive predecessor, Kadcyla, the Roche Genentech product. But we're not getting curative effects here. We are getting definite improvements in survival, definite improvements in progression-free survival, higher response rates. But the original studies which led to its approval in various indications generally compared it with chemotherapy. And it's a marked improvement over chemotherapy. It would want to be. And yet it's kind of, to me, it's kind of, I mean, you look at its first approval was HER2-positive breast cancer, obviously metastatic breast cancer. These were patients who'd received an awful lot of prior therapy, so they were reaching the end of the road in terms of anything that would work for them. It offered median progression-free survival of 16.4 months. Well, I mean, you wouldn't sneeze at that. That's not bad. And a 60% overall response rate, including, though, only a 4% complete response rate.
This is the problem with cancer. This is not an indication like a bloodstream infection or even a meningitis, where you have a 7 to 14 day course of an antibiotic that costs pennies and you are cured and you go home to your family at the end of it. A cancer diagnosis, if you talk to oncologists, they talk of at the end of treatment, you go into remission. They don't really say cure, because you never know. When I first started hearing that, I started thinking about glioblastoma. Sure, it's hard to treat brain cancer when it's an isolated site and getting stuff in there, and certainly with agents that were not approved or used off-label and then not very good. It was the survival with malignant glioblastoma is months. But this is someone's life we're talking about here after a breast cancer diagnosis, after a HER2 breast cancer diagnosis, but then after a number of therapies, number of lines of therapies as well. So actually, I think your earlier point of AstraZeneca doing that deal with Daiichi Sankyo, where they had to go out to the stock market and raise money to get money to do that deal.
Yeah.
You know, the business development and the people at AstraZeneca and the scientists and the clinical must have been working overnight together on that. Because as you also mentioned earlier, up to that point, the track record of drug antibody conjugates was not that good. And you would think, why would you want to put $1 billion in, let alone $8 billion into it? And you put it, but in HER2, it's shown a difference in HER2 positive. It's shown a difference that Kadcyla couldn't do over Herceptin in clinical studies. So I think we have to think of it as we're on a journey in cancer chemotherapy. It is a couple of steps up to an eventual cure, which is much better than where you were in the hallway with horrible cancer chemotherapy. Actually, that some of these agents are conjugated to antibodies with. So drug antibody conjugates, are we done yet? Probably not.
No,
we're not. The question is, how much more is there we can eke out without another advancement in either, as you talked about, antibody engineering? I think that's largely done. I mean, we've known the sequence of fully human antibodies for over a decade now. So if there was right site to know to optimize it, we would know it by now. Linker chemistry, that seems to be going by leaps and bounds. The toxic payloads, I think they're largely known. So we're not done yet, but are we over halfway? Are we 3/4 of the way along the development? And there are other products coming out. I mean, the bispecific antibodies, as I mentioned previously to you, certainly multiple myeloma, looking to eat the lunch of any drug antibody conjugates. On the other hand, there's new drug antibody conjugates in development from AbbVie targeting c-Met that have a wide range of potential cancer indications. So perhaps, I don't want to quote one of the fiancés of one of the Trump sons, but perhaps the best is yet to come in drug antibody conjugates.
I have no idea who you're referring to there, but multiple myeloma throws up the specter of another Lazarus-like ADC. This is GSK's Blenrep, or belantamab mafodotin, I believe is the correct pronunciation.
I mean, if you think for what Enhertu has done for the reputation of drug antibody conjugates, then Blenrep initially came along and swore in front of the TV screens on a live program and did the opposite for the reputation of them.
But see, this is very interesting because for people who might know the backstory, it got approved in 2020 in multiple myeloma, I think as a 4th-line therapy or 5th-line therapy, even for people who'd received, again, an awful lot of prior therapies, sadly at the end of the road. And this was a kind of a last-ditch effort to extend their lifespan. And it targets B-cell maturation antigen, which we have previously encountered because there are 2 CAR T-cell therapies targeting B-cell maturation antigen in multiple myeloma.
Which in some patients, as you pointed out to me, are curative.
Yes, but GSK withdrew this drug from the market about a year ago, this time last year, because it was being basically outcompeted by other products.
And also it failed a confirmatory phase 3 study.
Yes, but... And here's the big but. Just at the end of November, they reported it did better than Darzalex, which is an unconjugated, just regular monoclonal antibody, which Johnson & Johnson has developed with Genmab. It targets CD38 and it's a multiple blockbuster drug combined with other agents. And again, it is improved further up the line because it's been around the market longer. So GSK is now claiming that it has outperformed Darzalex. And it's going to obviously run hard for, they've withdrawn the original marketing approval. So I don't know if they'll have to go all the way back to a BLA all over again, or whether they'll have to do just seek a line extension on the original BLA. But that would be a very interesting story if that actually holds up.
Yeah, I think they should trade name from Blenrep to something like Lazarus Rep, because the eye toxicity was completely savage and the clinical data was not that good. And then new CAR Ts and bispecifics, as well as the standard naked antibodies, have come along in multiple myeloma to snatch their market away while they were sleeping and the FDA was taking them off the market. And then to come back again from that, it's got to be a good thing because, oncologists, particularly hematological oncologists talk about the sequencing and the phasing of different therapies for the same patient, or whenever they get to see them, the later line patients. So it adds another step, and that regimen, we talked about patients not actually being cured. It may keep, and this is again another phrase that oncologists use, is they want to keep people in remission for as long as possible. So if that is sequencing from different therapies to do it, even though as they sequence along, therapies fail, the toxicities are increased, patients live 6 months or to the next, and they always think, and I'll bet they're not thinking, that perhaps the next drug antibody conjugate that comes along for multiple myeloma will be invented while I'm still in remission for this one. So you never know. You never know what's going to happen. But it is a Lazarus-like regeneration of an antibody that people have forgotten. But we, as you said, with Mylotarg, with ADCs, but even drugs like antibodies like Tysabri, it's not the first time we've seen drugs come back from the dead after toxicity.
Yeah. And it's a good thing too. I mean, very often you see drugs that fail in a clinical trial and they get shelved, terminated, dispensed with. And we all know that, okay, they couldn't generate the evidence needed to make a certain commercial case. Scientifically, it's never the be-all and end-all at all, but they would be unwilling maybe to commit any more resources to further exploration. But because it's impossible to explore every single possibility attached to a particular drug in any particular setting, there's just too much complexity going on all the time.
And I understand that point, actually, because if you're a commercial person, like I used to be in a pharmaceutical company, even if it's not your first indication, and in oncology it never is. In oncology, you don't know the full toxicity, the side effect profile, the tolerability profile of a drug. And typically they start off, as you mentioned earlier, in the last line of therapy, rescue therapy for patients, and then they trickle their way back up. But the commercial people will be eyeing, as Merck did and Bristol Myers Squibb did with anti-PD-1 antibodies, the first line lung cancer for those products. And because it's such a big market compared to the others, and the pricing arguably doesn't change in that period. So you have that aspect, but when toxicity comes in and throws that spanner in your engine, then you're restricted to either a later line. But the FDA has done some wonderful things. One of them is to bring in this REMS process, risk mitigation strategy, where it decides much more stringently what patients can receive the drug, how they should be monitored and kept an eye on. So it allows drugs that would have been taken off the market and stay off the market to come back again in a highly managed way that allows it to treat more patients.
Yeah, true. But coming back to your point too, though, about whether we're beginning to reach the end of the road in terms of the possibilities in ADCs, I mean, the one question I'd raise there is that the actual range of tumor antigens that have been targeted by approved products is still narrow. I mean, we've got Mylotarg CD33, Adcetris is CD30. There's a bunch of them target HER2, a bunch of them Pfizer's Besponsa, and AstraZeneca's Lumoxiti, which has also been withdrawn actually, and that's actually dead and buried now at this stage. They both targeted CD22. Genentech has another product, Polivy, which targets CD79B for diffuse large B-cell lymphoma. Adcet from Seagen or Seattle Genetics and Astellas, Nectin-4, Trophoblast cell surface antigen 2, TROP2, that's Trodelvy, Immunomedics, acquired by Gilead Sciences for $21 billion, possibly overpaid there, I think. So epidermal growth factor, receptor tissue factor, CD19, that's HER2 folate receptor alpha, that's it. I mean, that's what we have. In clinical trials, there's an awful lot of the above, but there's a few other ones, CD123, CD25, B7H3, ROR1, etc., etc. So I think there's a lot more just antigens that are already targeted by naked monoclonal antibody therapies that need to be tested with ADCs as well.
Well, yeah, but some aren't applicable. I think you have to remember that it's not a panacea, antibody-drug conjugation. It's not a panacea. You can't just take every antibody that's ever been used in cancer therapy and apply a linker, put a payload on it, and it's going to give you better results. That didn't happen for years in HER2. Now, you can understand it in HER2 because you've got a defined cancer cell that the antibody will be attached to, and it will be taken into the cell, and a payload will be released, and you'll get toxicity for that tumor cell. Not all antibodies are there to lyse the cells. I mean, you won't see on your list VEGF antibody-drug conjugates. And you thought you would have seen PD-1 or anti-PD-1 drug conjugates, but they're more used for signaling. I'm surprised, actually, you don't see CD20. But, I mean, CD20 antibodies do a really good job as they are. So you don't need that sledgehammer. And we talked about toxicity, certainly with Blinrep. You don't need that sledgehammer to crack that nut. Antibodies are good enough as they are.
Yes, that's true. Then we come to the megabuck deals. Pfizer is paying an awful lot of its COVID products to acquire Seagen. And Seagen, previously known as Seattle Genetics, I guess has been the sort of, if you like, quote unquote, Genentech of ADCs. They really have kind of carved up in space, developed an awful lot of foundational technologies, brought several products to the market, bits of their technologies and other people's products. They really have been a leader. And they've accepted a bid for $43 billion, which is a very, very hefty buyout price. And it's a huge commitment from Pfizer. They really are obviously going to pile in here strongly. I don't know if you've given any great thoughts about whether they've done the right thing. Is this a good use of their resources?
Well, they had to do something after the pandemic. They needed to do something. They had the cash. And at the same time, they had a flagging oncology franchise compared to most other big pharma companies. So they needed to do something to address that. And you could also say that AbbVie earlier or at the end of last week did something similar, or that prompted AbbVie to do the same thing, or that confirmed Pfizer's purchase of Seattle Genetics. Because if you're out Christmas shopping for something and you're a pharma company, then you go for an antibody-drug conjugate company. Well, I don't think they're going for the platforms. I think both Pfizer and AbbVie have a post-multi-blockbuster product scenario to think about. And they have nascent, perhaps floundering, certainly in Pfizer's case, floundering oncology franchises. So if you wanted to just kickstart that with some more sales, and it doesn't matter what those sales are, but if you can hang a drug antibody conjugate platform on that, then that makes a good story. Then yes, I think it's more to do with supporting those oncology franchises within those big pharma companies and what happens after their blockbuster product go away than it is for saying, oh, well, we've got to do an AstraZeneca. If you look on the price to sales ratios of those transactions, again, AstraZeneca did a fantastic deal. And as you've gone on further along, sure, Seagen's sales were much higher than Immunogen's, which AbbVie is buying. So the price per sales for that deal, it's actually getting higher. So you're paying more for less, effectively. It does make me wonder whether we've seen the top in those transactions. If those two, Seagen and Immunogen, were bought just to bolt on sales, then it's got nothing to do with antibody-drug conjugates. It's just that we need to do this. And the proof, as you said, in the pudding is that the last people you would think of to do an antibody-drug conjugate acquisition was BioNT or Bionex. They're an mRNA company. What on earth are they doing it? Well, you can sort of see the corporate development people thinking, we've got this load of cash, we need to do something with it. And I think the Novo in particular has done an awful number of cardiovascular deals, little things, bolt-on things, new technologies. And shareholders will ultimately thank them. Not all of them will work out for it, but shareholders ultimately thank them for them because you didn't get a load of cash, you didn't sit on it and do nothing with it. And at least Pfizer and AbbVie and BioNTech are doing things with their windfalls.
And the reason though I would think that BioNTech, it's justifiable that you see them as an mRNA company and here they are doing this. I think they've always seen themselves as a cancer company.
Yeah.
And that the original company set out with multiple technologies under the hood. And the mRNA one was the one that progressed because of the Genentech deal about a number of years back. That was the one that really brought them to prominence. But they've always had quite a lot of arrows in the quiver. And it's interesting to me that they've done 2 deals in ADCs this year. And also interesting that both companies that they've done the deals with are in China, Medilink Therapeutics and Duality Bio. There's seems to me quite a lot of innovation coming out of China in ADCs.
Well, you could look at it that way, certainly not just on ADCs. I mean, AstraZeneca went to China for their GLP-1 receptor agonist for obesity. So after you poured over everyone else and reject, I mean, again, it might be a downside to antibody-drug conjugates. Once you poured over every company in Europe and the US and perhaps other places, and then you get your people on a plane to China, you then look there. I'm not scraping the bottom of the barrel, but this is perhaps not the first port of call for business developers, but it might be the last port of call.
Yeah, true. Because AstraZeneca has also done a deal in China this year with Kim Biosciences. Well, that's a US company, but it's a joint venture between two Chinese companies, and that's for a product that targets Claudin 18.2 in gastric cancer. So there's a huge amount of activity here. And it's still early days. I mean, the Enhertu, if you like, is the sort of flagship product of the field now. It was only approved in 2019. It's going to hit double blockbuster status this year. So it's still it's a mature field, I guess, a modality, but it's early maturation, I would argue.
Certainly in Enhertu's case, if you think poor old Roche with firstly Herceptin, having biosimilar competition, then Kadcyla coming to the market and not being that bigger, and then suddenly Enhertu with the Japanese company, where did they come from? You know, comes out and starts eating their lunch even more than the biosimilars are. So we talked about, have we seen the best to come yet? I don't think we've seen the best in terms of the penetration of Enhertu into whatever lines of HER2 positive breast cancer. But I mean, how big can it get? Can it go to first line? Possibly, but would you get an antibody drug conjugate with the toxicity we talked about, even though they're better because of linker technology, in first line metastatic HER2 positive breast cancer? I don't know, but it's on the way, right? And you'd never see that with Kadcyla.
And I think even though Kadcyla this year is still vying with it for sales, it's done $1.7 billion in sales so far. It's quite extraordinary. But to me, the point you raised about the toxicity is very real. I mean, again, in the early clinical trials of Enhertu that led to its original product approvals, like you actually had 2.6% of the patients died. You had a large percentage, 9% of the phase 1 and phase 2 trial patients in HER2 positive breast cancer developed interstitial lung disease or pneumonitis. I mean, that's a very serious life-threatening condition.
And that is, I mean, that particular toxicity is a function of the payload that they use there, just as ophthalmic toxicity is part of Blenrep's profile.
I think that, to me, without kind of having delved too deeply into it, I think there's still huge possibilities on innovation in terms of balancing the systemic toxicity or the off-target toxicity with the on-target efficacy. I don't think that is a solved problem yet, and I think there is huge scope there for more innovation, which would give you a much better therapeutic window. I would think with the ADCs, to me, I don't like the term magic bullet at all. I think it's a metaphor that's been overused, and it goes back to Paul Ehrlich, I think, doesn't it, in the late 19th, early 20th century. They're not bullets. We are, yes, trying to kill a cancer, but we are also trying to preserve the life of a patient, and that's much more important. And
their quality of life as well, you know, even on therapy. So precisely.
So I think that just at a high level, and I know there's smart people working in this problem in multiple labs around the world, I think there's an awful lot more innovation to come in terms of optimizing the tox piece with the potency piece.
Yeah, I think you make a really good point. And there was me leading listeners on the line to saying, have we already seen the best of antibody-drug conjugates? Well, on the other hand, if you're a young listener thinking of going into science, do medicine or chemistry, do your master's or your PhD in drug antibody conjugate linker chemistry, and you'll probably quite easily get a job at the end of it.
Yeah.
But more importantly, you could be responsible for the next Enhertu.
Well, exactly. Make a very useful contribution.
Yeah.
Because I think maybe the way we could sum it up is the best is yet to come with ADCs, we hope.
Yeah. Yeah, absolutely. Very good. Very good.
Yeah. Okay.
You don't need to listen to this the rest of the time. You just need to listen to the last 2 seconds.
Until the next time, Andy. Thank you.
Cheers, Cormac. Bye-bye, everyone.